basal ganglia. So basal ganglia, it means that it's on a base. It means it is on a base, and ganglia means just bunch of neurons, collection of neurons. We call it ganglia. So basal ganglia, the lucky number is 5. It means 4 plus 1. So 4 nuclei, 4 ganglia are inhibitory and 1 is excitatory. So what are those 4 nuclei? The first is caudate, second is putamen, third is globus pallidus, and fourth is substantia nigra. Those 4 ganglia are inhibitory in nature. It means they work on GABA. And subthalamic nuclei, subthalamic ganglia is excitatory. It means they work on glutamate. This is your brain. Caudate, the word caudate, cauda means tail, cauda equina, tail of a horse. So caudate is a C-shaped nuclei with a tail. Putamen is a pit, so shell pit, lies like a shell. Globus means sphere, pallidus means pale. Why is it pale? It's pale because it's myelinated, it's highly myelinated. So globus pallidus is externa and interna, and interna and externa. Note that interna is the output nuclei and externa is not the output. And caudate plus putamen, it gives us something called a striatum, and this striatum is the input nuclei. And substantia nigra has 2 parts, part reticulata and part compacta. So compacta means it's really tightly packed and it's really pigmented. Why is it pigmented? Why is it pigmented? It's black in nature, and that's why we call it nigra, because it's black. And parts reticulata, it's a reticulum, it's a mash of gray and white matter. Gray matter nuclei passing through white matter bundles, So for example, this is gray matter nuclei, and then we have a lot of white matter bundle fibers, okay? So pars reticulata, GABA, pars compacta, dopamine, works via dopamine. Dopamine is inhibitory for prolactin. So this is pars compacta. And subthalamic is excitatory, it works via glutamate. It is excitatory. So these are like basic parts. It's not anatomy, it's really just the glimpse, okay? What about physiology? Well, physiology is that basal ganglia works via planning, in cognition, in inhibition of involuntary and voluntary. Basically, it lets you stop, it lets you take rest. Basal ganglia. If you want to sit on a stool, so what happens is that basal ganglia inhibits all the voluntary and involuntary movements, and if your basal ganglia is messed up, you won't be able to sit idle. So basal ganglia lesion leads to involuntary movements. So this is basal ganglia. So that's the basic idea, that's the function. Let's about pathways. So we know that it has one input and one output. So input is striatum and output is globus pallidus interna, okay? So this is globus pallidus, this is externa, this is interna, and this is the output one, and caudate putamen, striatum is the input one. So cortex, this is, for example, how this works is that this is cortex. So fibers come in to the striatum from the cortex, and then eventually all the processing is done, and the output is this externa, GP interna, and the output goes back to the cortex, and then the processing is done. That's how more or less the physiology is of basal ganglia is. Let's talk about the pathway exactly. So it has 2 pathway. It has one direct, one indirect. Indirect pathway is to inhibit the movement, direct pathway is to facilitate the movement. So if your direct pathway is messed up, it means no movement will be done. Ataxia, I think, ataxia. And if your indirect pathway is messed up, you won't be able to stop moving. So lots of involuntary movements, okay? So let's talk about direct pathway. Direct pathway starts from 1 to 2, that is the cortex to this striatum, okay? Striatum putamen, okay? And this is how the direct pathway is, just direct. And indirect is like this. I will tell you how. So cortex give fibers to striatum, that's the input of basal ganglia. Then this striatum the striatum is negatively inhibitory. It works on GABA. So striatum produces GABA. GABA then works on GP interna. It inhibits GP interna. Interna is the output one. So if GP interna is inhibited, it means it will not produce anything. It will not inhibit this ventro-anterior nucleus, and this ventro-anterior nucleus, this ventral anterior nucleus of thalamus, is responsible for the movement. So if your ventral anterior nucleus is inhibited from GP, how it's done? It's done via indirect. So the thing is, we have to deal with this nucleus, okay? Ventral anterior nucleus. This nucleus, ventral anterior nucleus, is responsible for movement, okay? So if you inhibit this nucleus, there will be no movement. If you facilitate this nucleus, there will be movement. This is facilitated by how is this facilitated? Basically, it works via GABA, right? If you put GABA inside it, it will be inhibited. If you don't put GABA inside it, it will be facilitated, basically. And it works via, like, GABA is provided via indirect pathway. What happens in indirect is cortex give glutamate to the striatum. Striatum is activated. Striatum is GABA. It inhibits what? It inhibits GP externa, okay? And GP externa further inhibits this subthalamic, okay? And if the inhibition is lost, and if there is inhibition lost, then the GP externa will not be, activated, and what happens that this will be inhibited. This ventro-anterior nucleus is inhibited. What I want to say is that cortex gives glutamate to striatum, striatum gives GABA to interna, and interna is inhibited, interna stops working, so the pathway stops here. If the pathway stops here, it means ventral anterior nucleus, this VAN, is working, and if VAN is working, then we have movement. And indirect, we have GABA stops interna, and then further GP is not stopped, and then GP interna produce GABA on VAN, and its VAN is inhibited now, and then there will be no movement. So indirect pathway for stopping the movement, and direct pathway for facilitation of the movement. What about substantia nigra? Well, substantia nigra produces something called as dopamine via, via pars compacta, not reticulata. Sorry, pars reticulata is this mesh, okay? It's like this mesh, and these are like gray matter here everywhere, and this reticulata is the output nuclei. And what happens is that this pars reticulata is, via D2 receptor, it inhibits this striatum, okay? So that's what happened, okay? So via D2 receptor, D1 in direct, D2 in indirect, and this D2 receptor inhibits the indirect pathway. So if D2 receptor is inhibiting indirect, it means it's promoting direct, okay? And if it's substantia nigra via D1, it facilitates, okay? So movement is done. So dopamine facilitates direct and indirect pathway. That's why you dance. That's a good explanation. Whenever your dopamine is high, you feel like dancing, because a lot of dopamine is facilitating striatum, and then striatum is further inhibiting GP interna, and then GP interna further not inhibits this ventral anterior nucleus. It's not a very complex pathway, but I think my explaining is very poor. I'm not able to explain it very well. It means I am not able to understand it very well. But I think this pathway is very simple. If you read it yourself, just take a screenshot and try to read it yourself, and you will be able to understand what's happening really. Okay, let's talk about the disorders. So disorders of basal ganglia are Parkinson's, and I think that's it. So Parkinson's, it's great. So it's a disorder of GABA. It's a disorder of dopamine, not GABA. GABA is for different one. Sorry. So disorder of Parkinson. If you have less dopamine in substantia nigra, imagine you don't have dopamine here. Okay. So there is no facilitation to striatum. So if there is no facilitation to striatum, it means it will not be activated. If it's not be activated, it means there will be no GABA on interna. If GP interna is activated, it means that it will put GABA on VAN, so there will be no movement. So less dopamine, pars compacta. Clinic feature bradykinesia. It means slow movement, very slow movement, very bad movement. Tremors, it will be pin rolling tremors, resting tremors, okay? Whereas in cerebellum, it was, if you tell the patient to move, he keeps on moving and the tremors increase while he moves more and more. But in substantia nigra, in Parkinson's, the tremors are there when the patient is on rest. Okay, so this is what happens, in Parkinson's. Okay, the treatment is L-DOPA. Why we give L-DOPA? Because dopamine cannot cross brain-brain barrier. Okay, what's not big? It's blood-brain barrier. It's not it's blood-brain barrier, and that's why L-DOPA we give, because L-DOPA can, it can effectively jump the BBB. I will tell you how it jumps. Very easy. It's basically, there is like guard cells of BBB. L-DOPA can inhibit these guard cells of blood-brain barrier, and then it can like jump the blood-brain barrier. So that's the story of L-DOPA. What about surgical therapies? Surgical therapy can be ablation and deep brain stimulation. Ablation is just removal. So you can just remove this ventointerior. It's not really ablation. It's not really removal. It's ablation. Ablation is literally a little bit different. So in thalamus, you have ventral anterior nucleus. You know that VAN is for movement and VLN is also for movement. So these are ablated, and then basically you can move well because these are inhibitory nucleus. It's a GABAergic nucleus, right? This is a GABAergic nucleus. If you stop them, you can move well, right? So symptoms elevated. What about deep brain stimulation? Well, you stimulate basal ganglia, subthalamic nucleus. What is subthalamic nucleus? I just told you here. Subthalamic nucleus is a excitatory. If you excite this, if you stimulate this subthalamic, you'll be able to move well. You'll be able to, move pretty well, improves your movement. So yeah, this is how it's done. What about this disease called Huntington's? So yeah, this was the disease. This is a GABAergic, I think. This is a GABAergic, disease. I think this is a GABAergic disease, right? The loss of GABA. So Huntington disease. So we have something called as Parkinson's. Parkinson's is because of less dopamine in nigra, and GABAergic is due to Huntington gene. So there is something called as Huntington gene. This gene is neuroprotective, very important. It's anti-apoptotic. What is anti-apoptotic? Well, apoptotic is a process of cell dying and cell suicide, basically. So it stops cells from killing themselves. So anti-apoptotic and neuroprotective. It protects the neurons from radicals and everything. And so Huntington, we need this, and it produces something called as brain-derived neurotropic factor. It's very important for the development of it's very important for the plasticity of your neurons. So basically, if this is messed up, what happens is that mutation of Huntington gene happens, and then you have something called as trinucleotide repeat. It looks like very complex, but it's not. It gives us something called as CAG. What is CAG? Is that repetition of 3 nucleotide DNA sequence, and we get something called as CAG. What is cytosine adenine guanine? So what happens that the repetition of 3 nucleotides, right, leads to increased polyglutamine levels. Okay, and if these polyglutamine level, that glutamate is excitatory, right? So what happens in Huntington is that this glutamate levels are through the roof. So polyglutamine levels are through the roof, and then it translates to proteins. So what happens in polyglutamine levels? Translated proteins has a stretch of many glutamines. Okay, the longer the polyQ stretch, the more toxic and misfolded the proteins get. So that's why you don't want a lot of polyglutamine levels. The excitotoxicity increases. More glutamate equals to more NMDA, EMPA stimulation. We will read about them right now soon. And what happens is that NMDA gets stimulated, we have calcium influx, and then we have stroke. Not from stroke, basically a lot of calcium, it results in a lot of further downstream processes. Okay, so calcium is like a signaling molecule. Okay, if you have a lot of calcium inside your cell, it means it's a signal to a lot of cells that something is wrong. So a lot of calcium influx via NMDA or EMPA receptors, and then you have stroke or Huntington or cell death. Okay, so loss of GABA. Okay, loss of GABA releasing from the cell from caudate nuclei striatum. Okay, loss of GABA. What happens is that here we know that this is the striatum, right? This is the input one. So if in this pathway, imagine this is the pathway, right? and then this is the striatum, right? And you have a disease called Huntington, right? And this in the Huntington, you have something called as poly Q, right? And this polyQ leads to CA influx, and this CA influx leads to loss of GABA. And GABA is the most important neurotransmitter in this striatum, because GABA is the only one, depressing the cell, depressing the GABA basically just reduces the electronegativity of the neurons. Neurons are minus 70 millivolt. What it does is it jumps the neurons to maybe minus 90 or even minus 1 or 10. So we don't want that. If your cell is really negatively charged, it's really depressed, and then you need more and more positively charged ions to excite the cell. So again, loss of GABA happens, releasing from the caudate nucleus. If your caudate nucleus, this striatum is messed up, for example, this is stopped, right? This stops working right now. So what happens is that there will be no inhibition of GP interna. If GP interna is not inhibited, it will keep inhibiting this ventral anterior nucleus. If ventral anterior nucleus is this ventral anterior nucleus is very excitatory. If this is stopped, it means okay, you will not have any movement, because glutamate, it excites you. It's excitatory nucleus, and glutamate stops getting released from this VAN. So if this is messed up, in Huntington. So important thing is that in Huntington, striatum is messed up. And important thing in Parkinson is that your substantia nigra compacta is messed up, right? So that's the whole story. Pars compacta is messed up in this Parkinson's, and the striatum is messed up in Huntington. So that's the whole story of it. That's the whole gist of it, right? So memory abnormality also happen. Cognition is affected, because we know that basal ganglia plays a big role in in this cognition. Caudate nucleus, this one, and caudate nucleus leads to cognition. So that's the whole story of it, right? What about involuntary disorders? Well, involuntary disorders are type is chorea. What is chorea? Chorea comes from the word choreography. So any sort of movement is chorea. Atheritosis is it's a slow movement. Okay, so slow writing, atheritosis, and there's something called as hemibolism. It's basically half part of your body. It contracts very fastly. It looks like a violent contraction of large joints. Okay. And it's not hemibolism. It's sudden violent contraction of a large joint. And what happens that chorea happens because of caudate nucleus, and we know that chorea is because of the movement, and we know that this caudate nucleus, the striatum, right? So striatum is responsible for the movement, right? So if there any sort of movement disorder, caudate can be very much be responsible for this disorder, right? And this thing called atheritosis, it's slow writing. It means it's some sort of bradykinesis, right? So it's some sort of bradykinesis, and it happens in Parkinson's. So globus pallidus is affected here, right? Snake charmer, because he moves very slowly in front of the snake. That's just way of explaining it. Hemibolism is sudden violent contraction of large joint, flinging and ballistics, and subthalamic nucleic. Okay, so if you have some problem in subthalamic nucleus, you will have hemibolism. Okay, so that's the whole story of basal ganglia. It's a very simple topic, but you have to really understand this neural pathway very well. If you don't understand this neural pathway very well, you will not be able to understand basically what is the concept of it. Okay, because we need to understand striatum is actually, it's very inhibitory. So it inhibits this VAN. Okay, if it's not inhibited, okay, because VAN is excitatory, right? So you need to understand this neural pathway. So I would love to actually study it again and able to explain you better. So have a nice day. Basal ganglia, that's it. 4 plus 1, 4 are negatively charged, not negative charge. Inhibitory plus 1 is the excitatory, that is glutamate.